Good day, everyone. I am Farhanah Rangiris and I'm going to present this review article entitled Prokaryotic Cytoskeletons, Protein Filaments Organizing Small Cells, written by James Wagstaff and Jen Lowy. Well, before we proceed, I advise you to listen attentively to what is being discussed because some parts were obviously not presented on the slides. But note that some of the key terms are defined on the left side of the slides. Most, if not all, bacterial and archaeal cells contain at least one protein filament system. Although these filament systems in some cases form structures that are very similar to eukaryotic cytoskeletons, the term prokaryotic cytoskeletons is used to refer to many different kinds of protein filaments. Cytoskeletons achieve their functions through polymerization of protein monomers and the resulting ability to access length scales larger than the size of the monomer. Prokaryotic cytoskeletons are involved in many fundamental aspects of prokaryotic cell biology and have important roles in cell shape determination, cell division, and non-chromosomal DNA segregation. Some of the filament-forming proteins have been classified into a small number of conserved protein families, for example, the almost ubiquitous tubulin and actin superfamilies. To understand what makes filaments special and how the cytoskeletons they form enable cells to perform essential functions, the structures and functions of cytoskeletal molecules and their filaments have been investigated in diverse bacteria and archaea. In this review, data are brought together to highlight the diverse ways that linear protein polymers can be used to organize other molecules and structures in bacteria and archaea. However, this presentation will cover only two of the various types of protein filaments, namely the tubulins and actins. Tubulin homologues are widely distributed in bacteria and archaea and are involved in many different cellular processes. The dynamic properties of tubulin filaments are often used for cytomotive functions, that is, to push or pull other molecules in the cytoplasm. Tubulin superfamily members share a distinctive globular domain composed of two independently folded but closely associated subdomains that are separated by a core helix, the amino terminal GTPase domain and the carboxyl terminal activation domain. Polymerization occurs through the association of a GTP-bound GTPase domain from one monomer with the activation of the next monomer, forming a catalytically active GTPase site around a GTP molecule at the center of the intersubunit interface. Many monomers associate head to tail in this way to form a one subunit thick filament known as a protofilament. The cell division protein FITZ was the first component of a prokaryotic cytoskeleton to be identified. FITZ has the common tubulin bipartite globular domain at the end terminus. It is localized near the membrane at feature division sites in almost all bacteria and most archaeal phyla and forms a ring-like structure known as the Z ring, which contracts during the cytokinesis. In bacteria, FITZ is among the first molecules to arrive during the assembly of a poorly characterized macromolecular complex known as the divisome, which incorporates many of the enzymatic activities and other functional modules that are needed to carry out cytokinesis and to remodel the cell wall. Although the central role of FITZ in cell division of bacteria is well established, it has been better characterized using new light microscopy techniques. Higher resolution imaging of the Z ring in live bacteria suggests a new model for FITZ function whereby relatively short FITZ filaments treadmill circumferentially around the division plane to drive cell wall remodeling divisomes around with them. The mechanism of FITZ function in archaea remains almost totally unexplored, although it is known that many euryarchaeal genomes encode two FITZs termed as FITZ1 and FITZ2, with both of them involved in division. Some magnetotactic bacteria encode an additional FITZ-like protein termed FITZM that does not have the C-terminal extension that is required for interactions with many partners. Although the biological role of FITZM is unclear, it apparently copolymerizes with FITZ and is therefore recruited to the Z ring. FITZM filaments have also been implicated in redox-dependent regulation of magnetosome biogenesis. A distinct group of tubulin superfamily genes, namely SETZ, are found in some euryarchaeal genomes in addition to division-related FITZs. Their sequences diverge from both eukaryotic tubulins and bacterial and archaeal FITZs, but they share features with each. The group was renamed SETZ after the prototypical member that was characterized in this study, the tubulin-like protein SETZ1. SETZ1 is required in vulcanized species for the differentiation of irregular plate-shaped cells into a rod shape that is essential for efficient swimming motility. SETZ1 forms a dynamic cytoplasmic filaments that are required for cell shape determination. However, the mechanism by which SETZ proteins modulate cell shape is unknown. The localization of the protein was shown to be related to membrane curvature. TubZ proteins are a diverse but likely monophyletic group within the tubulin superfamily. TubZ proteins are predominantly encoded on rapidly evolving non-chromosomal DNA, for example, in bacterial plasmids and in bacteriophage genomes. TubZs include the type 3 systems of plasmid segregation and the FuZ subfamily of bacteriophage proteins that position viral DNA and virions within the host cell during viral assembly. Although we do not know whether or how FITZs and SETZ protofilaments might associate to form higher order structures in cells, in the case of TubZ, several parallel protofilaments associate to form the functional cytoplasmic filaments. TubZ proteins have a C-terminal extension that reaches along the protofilament to the next monomer and is crucial for robust filament formation and for wild-type filament dynamics. Other members of the tubulin superfamily are found in bacteria and archaea. These are identified by the highly conserved sequence motifs of the GTPase domain. A pair of bacterial tubulins termed B-tubs A and B or B-tub AB that are more similar in sequence and structure to eukaryotic tubulins than to FITZs have been found in addition to FITZs in several Prothyscobacter species genomes. B-tub genes were most likely acquired in a horizontal gene transfer event from a eukaryote to a Prothyscobacter species ancestor. It forms four-stranded mini microtubules in vitro, exhibiting a seam and dynamic instability, which are two hallmarks of eukaryotic microtubules. Some members of the Thaumarchaeota within the archaeal tag superphylum encode a eukaryotic gamma-tubulin-like protein called R-tubulin. It isn't clear whether R-tubulins are the product of a horizontal gene transfer event or represent a vertically inherited ortholog of gamma-tubulin. More convincing putative ancient tubulin-like genes were found in recently published archaeal genomes assembled from the metagenomic sequence data. Two groups of proteins with homology only to the GTPase domain of the tubulin superfamily have been identified in bacterial and archaeal genomes. Both groups have been termed as FITZ1. It has been suggested on the basis of proximity of FITZ1 genes to membrane remodeling genes in genomes that these proteins are involved in membrane remodeling. It isn't clear how or whether FITZ1 proteins could form filaments because they lack a C-terminal domain homologous to the tubulin superfamily C-terminal active domain. actins, like tubulins, are versatile cytoskeletal components that are able to form robust cellular scaffolds and also provide dynamic properties that can be harnessed for cytomotive functions. MRB is the prototypical bacterial actin found in almost all wild bacteria with elongated shapes. It is essential for normal cell wall synthesis in this organism, and it is responsible for organizing a multi-enzyme complex known as the elongosome. In some bacteria, MRB filaments are used for functions independent of cell wall patterning, for example, as a cytoplasmic component of the gliding motility machinery of Myxococcus species. Archaeal actins whose closest homologous are MRB have been identified but remain unstudied. The second actin superfamily is termed as FITZ-A, which is an unusual bacterial actin. Crystal structures and negative electron microscopy reveal that FITZ-A has a recognizable but unusual actin fold that can polymerize to form actin-like protofilaments. FITZ-A and FITZ-Z copolymerize in vitro to form a variety of dynamic structures, and they can cooperate to constrict liposomes from the insides. Fourth of the actin superfamilies is termed as MAMK, which forms filaments in magnetotactic bacteria that are used for the alignment and segregation of magnetosomes. It forms cytoplasmic filaments that connect ordered chains of magnetosomes. MAMK filaments are right-handed, parallel, and two-stranded with juxtaposed subunits. In addition to the three well-studied chromosomally encoded bacterial actins previously mentioned, there are a large number of diverse plasmid-encoded and bacteriophage-encoded actins. Although the abundance and diversity of this group of bacterial actins like proteins or ALPs were not recognized until recently, one member of the group has been the subject of study for many years, namely the PAR-M protein, which is prototypically found on the E. coli R1 plasmid. PAR-M forms filaments and functions for type 2 segregation system, which confers stability on their host plasmids by actively segregating plasmid copies into daughter cells at division. A number of ALPs have been partially characterized. Such investigation suggests that there is a substantial diversity in the filament properties and functions to be found among the ALPs. One archaeal actin, namely TAZ0583 from Thermoplasma acidophyllum, is probably the result of a horizontal gene transfer of a bacterial ALP into an archaeum. Lastly is the superfamily crenactin, which serves as an evidence affirming that eukaryotic actin architecture is ancient. This family is found was found in members of the Thermoprotealis class of the phylum Crenarchaeota within the tax superphylum. Thermoprotealis have no known filament or membrane remodeling system except for crenactin, and immunofluorescence microscopy data suggest that the protein has a cytoskeletal role. Over the past 25 years, it has emerged that cytoskeletons are important and often essential cellular components of bacteria and archaea. The protein filaments that form these cytoskeletons are useful because they organize cells at length scales larger than the size of their constituent monomers. A recent surprise has been the discovery of protein filaments that organize processes using a limited number of monomers in dynamic ways. It appears that bacterial FITZ-Z and MRB, the founding members of the prokaryotic cytoskeleton, both function mainly through short filaments that organize peptidoglycan remodeling locally. Such advances in knowledge mean we are reaching a point where our understanding of the biology of the major filament systems in well-studied bacteria is becoming comprehensive. By contrast, the basic understanding of the biology of filament systems in archaea remains poor. The study of these molecules, particularly in vivo, will be crucial for improving the understanding of archaeal cell biology and of how ancestral filament systems functioned. For both archaeal and bacterial filaments, the understanding of the molecular mechanisms by which these dynamic systems function is limited by a shortfall in vivo and in vitro structural studies. The only example where the researchers have an almost complete molecular explanation of function is the relatively simple PAR-M plasmid segregation system. The researchers await with excitement the additions to that list that will come in the next 25 years of prokaryotic cytoskeleton research. Again, this is Farhanah Rangiris, your presenter of today's topic. Thank you for listening.